Hello everyone. I, my name is Snehlata and I'm a PhD student and a PMRF fellow at the Department of Biological Sciences and Bioengineering. Today we will continue with the next lecture of the chapter Biomolecules. So till now we are studying about biomolecules, and in last lecture we studied about the enzymes. Hello. Hello.
हाँ, आप स्नेह स्टार्ट कर सकते हो। आप ऑडिबल हो और आपका स्क्रीन भी विजिबल है।
Okay, sir. Okay. Hello everyone, my name is Snehlata and I am a PhD student and a PMRF fellow in the Department of Biological Sciences and Bioengineering, IIT Kanpur. Today we will continue with the next lecture of the chapter Biomolecule. So in the last lecture we were studying about the enzymes, and in that we were seeing that what are the factors that affect the activity of the enzyme. And in that we studied about various factors such as pH, temperature, and substrate concentration. Apart from this, there was one more factor that affects the activity of the enzyme, and that was inhibitor. So the fourth factor that affects the activity of the enzymes are inhibitors. Now, what are inhibitors? So inhibitors are the chemicals. So these are the chemicals that either inhibit that either inhibits or reduce the enzyme activity So these are the chemicals that inhibits or reduce the enzyme activity. And if we talk about the nature of the inhibitors, so we have just seen that the nature is that they are chemical in nature. Now, how these inhibitors affect the activity of the enzyme? So they can affect the activity of the enzyme via various modes, among which the first one is either they can bind to, the inhibitors can bind to the active site of the enzyme. So till now we have seen that how enzyme works. So the substrate binds to the enzyme active site because the active site of the enzyme has the shape complementary to that of the substrate shape, right? And because of this reason, the enzyme binds to the substrate binds to the active site of the enzyme. But in the case of inhibition, the inhibitor also, it can bind to the active site of the enzyme and hence can reduce or inhibit the activity of that enzyme. The second mode by which it can affect the activity is it can disrupt the shape of the enzyme. It can disrupt the shape of the enzyme. And we have seen in the previous lecture that the shape plays a crucial role in the activity of the enzyme because of why? Because of the shape of the enzyme, the substrate binds to it, right? And the third is by binding to the, so the inhibitor can also bind to the enzyme substrate complex. So it can also bind to the enzyme substrate complex. Okay. So it's not like the inhibitor can only bind when the enzyme is vacant, but the inhibitor can also bind even when the substrate has already bound to the enzyme. Okay. And hence it can affect its activity. Now, based on this mode of action, there are two types of enzyme inhibition. So there are two types of enzyme inhibition. The first type is known as competitive inhibition. And the second type is known as non-competitive inhibition. Now, we will see in detail what is competitive inhibition and what is non-competitive inhibition. So the first one is competitive inhibition. So as the name suggests, the competitive inhibition, how it works. So these kind of inhibitors, so the inhibitors that work via competitive inhibition, these inhibitors compete with the substrate. Okay, they compete with the substrate for what? For binding to enzyme active site. For binding to enzyme active site. Okay. Now, when we were studying about the features of the enzyme, we saw that the enzymes are highly specific in nature, right? So the enzymes, one of the important feature of enzymes are their specificity, right? So they are highly specific in nature. And this specificity was because of the shape of the enzyme, right? Because of the shape of the enzyme. So how can an inhibitor bind to the enzyme active site? This is because these inhibitors, they have a similar shape. The inhibitors have similar shape as that of substrate. Okay. Because we know enzymes are highly specific in nature because of their shape, and they only allow those substrates to bind to the enzyme, those that have the shape complementary to the enzyme active site, right? But the inhibitors that have this similar shape can also bind to the enzyme, right? And these inhibitors act as a competitive inhibitor. Now, although these inhibitors have similar shape, but they are chemically different. So although they have similar shape, I'm not talking about the identical shape, but they have similar shape, but they are chemically different. So if we look at their chemical formula, that will be chemically different. Okay. So let's see what happens when the inhibitor binds to the enzyme. So in ideal case, when the substrate bind, this is how enzyme works, right? The substrate comes and bind to the enzyme via diffusion. And then what happens? Then a transient state form. And this transient state is called as enzyme substrate complex. Then this enzyme substrate complex, it further proceeds to form enzyme product complex. And then enzyme and product gets released from the enzyme, and then the enzyme recycled to so that a new substrate can come and bind to it, right? So this is how a typical enzyme works. But what happens when the inhibitor binds? So when instead of the substrate, the inhibitor that I am representing by I, so when the inhibitor binds, what happens? The enzyme inhibitor complex forms. But this enzyme inhibitor complex does not proceed further, and hence no product formation takes place. Okay, and that's how it reduces or it affects the activity of the enzyme. Now the question is how one can overcome this competitive inhibition. So in order to overcome the competitive inhibition, what we can do? So we can increase the competitive inhibition can be increased by increasing or can be overcome, right? Can be overcome by increasing substrate concentration. Okay, so this is substrate concentration. Now the question is how? So suppose initially, in the initial condition, what you have say the substrate, initially the substrate is 200 molecule and your inhibitor is also 200 molecules. Okay, then what will be the chance of the substrate to binding to the inhibitor? What will be the chance of an inhibitor to binding to the enzyme? So the chance will be 200 by 400, right? That is 1 by 2 or 50%. So out of 2 molecules that can bind to an enzyme, one can be inhibited, right? But if you increase the substrate concentration, say by to 1800, and your enzyme is still 200, now what will be the chance of an inhibitor to bind to the enzyme? It will be 1 by 10, right? So it will be like out of the 10 molecules, only one inhibitor molecule can bind to the enzyme, right? And by this way, the chance of the inhibitor to bind to the enzyme will reduce. Hence, we can overcome this competitive inhibition, right? So we can overcome the competitive inhibition by increasing the substrate concentration. And why I am saying so, you can understand this by this example. Now, next, let's see briefly about when we were studying about the enzyme features, we also, or when we were studying about the effect of substrate concentration on enzyme activity rate. So we came across two terms. The first term was Vmax and the second was Km. Now, in the case of competitive inhibition, what happens? In the case of competitive inhibition, we are seeing that is there any change in the enzyme number or shape? The answer is no. So it is unchanged, right? Because the inhibitor is coming and simply binding to the enzyme. So it is not affecting the enzyme number. The enzyme number before the inhibitor and after the inhibitor, all shape is same, right? So what will happen to the Vmax? So if you remember this graph, so there was graph between the substrate concentration and the velocity, that is the enzyme activity rate. So the graph was something like this. So graph was something like this, right? So with the increase in the substrate concentration, the enzyme activity rate was increasing, or the enzyme activity was increasing to a certain level, after which it becomes saturated. And this level is known as Vmax, right? This is known as Vmax. So if we see the Vmax is what? It is a maximum velocity enzyme can achieve. So even if the inhibitor binds or even if the inhibitor is there, it is not affecting the shape of the enzyme, and hence its activity will remain, the activity will remain unchanged, right? Activity will remain unchanged because the activity comes from the shape of the enzyme. Hence, the Vmax, if we talk about the Vmax, so if a substrate binds to the this enzyme, the Vmax will be same as that of initial, even in the presence of the inhibitor, right? So the Vmax remains unchanged. Now, the second thing, what about the substrate concentration? So we saw that in order to overcome this competitive inhibition, we had to increase the substrate concentration, right? So the substrate concentration have to increase. So we have to increase the substrate concentration. And why so? It is to overcome inhibition. To overcome inhibition. Now, if the substrate concentration is increased, then what will happen to the Km? So what I told about Km in the last class. So Km is what? So Km is that concentration of the substrate. So this is a Km. And this is a half Vmax. So what was Km? Km is the concentration of the substrate at which an enzyme can achieve half Vmax, half of its maximum velocity. So if we see, this graph is without inhibitor. This graph is without inhibitor, right? Now, if we talk about the inhibitor, the graph will look something like this. The graph will look something like this. So because it is not affecting the shape of the enzyme, the Vmax will remain the same. The Vmax of this will also remain the same as without inhibitor. But in order to overcome this inhibition, we will need higher substrate concentration, right? So if there is inhibitor, so this graph is with inhibitor. So if there is inhibitor, then in order to achieve the same half Vmax, you will have to use higher substrate concentration. So this will be your K-SM, that is in the presence of inhibitor. So the Km in the case of competitive inhibition, it increases. It increases. And why it increases? Because high or increased substrate concentration has to be used, right? Has to be used. So we understood where this graph, that because in order to overcome the inhibition, we need higher substrate concentration. So in order to achieve the same half Vmax, a higher Km value will be required, and hence the Km will be increased in the case of competitive inhibition. So the two important point is that in the case of competitive inhibition, Vmax remains unchanged while the Km increases. Now, let's talk about the example of competitive inhibition. So one of the important example is about the enzyme that is known as succinate dehydrogenase. The enzyme succinate dehydrogenase. Okay. The substrate for this enzyme is succinate. Okay. And the inhibitor for this enzyme is malonate. Okay. So the substrate is succinate and the inhibitor is malonate. Now, if you look at the structures of this succinate and malonate, so the succinate here, we can see it has an extra CS2 group as compared to that of malonate. But they have a similar kind of structure because the CS2 itself is not that big to make a very different shape of this enzyme substrate. So they both of this have similar structure. But if you look at their chemical formula, so although they have similar structure, they are chemically different, right? Because one has a extra CS2 group, so they are chemically different. So this is what I was talking about, a competitive inhibition. Because of this similar structure, this malonate can act as a competitive inhibitor for this succinate dehydrogenase, but because it is chemically different, it will not lead to the formation of the product. So this is one of the example of competitive inhibition. The second example is about the sulfur drugs. Now, what are sulfur drugs? So the sulfur drugs, these are the drugs that are used against the pathogenic bacteria. So these are used against pathogenic bacteria. Okay, pathogenic bacteria. Now, what does it do? So in the pathogenic bacteria, in any bacteria, we know in bacteria also they do respiration. Some bacteria do photosynthesis, and there are a lot number of chemical reaction that takes place in the bacteria as well. And for that purpose, enzymes will be required. So these sulfur drugs, they are designed so that they have similar shape. As that of bacterial enzymes. Okay, as that of bacterial enzymes. So if they will have similar shape, what they will do? They will act as a competitive inhibitor, and instead of bacterial enzymes substrate. So bacterial enzymes substrate. So the substrate for a particular enzyme. So these sulfur drugs will have the shape similar to the substrate that binds to the bacterial enzymes, but they will not lead to the formation of any product. And if the product will not form, then that product will be definitely required for the survival of the bacteria, and hence the bacteria won't survive if that product will not be given to that bacteria, right? So these sulfur drugs, they have similar shape as that of the bacterial enzyme substrate. By bacterial enzyme substrate, I mean substrate that bind to the bacterial enzyme. Substrate that bind to bacterial enzyme. Okay. So by this way, these sulfur drugs act as a competitive inhibitor again, and hence they are used against the pathogenic bacteria. So pathogenic bacteria are the one that infects our body. Okay. So this is all about the competitive inhibition. Now next thing that we'll study is about the non-competitive inhibition. So what is non-competitive inhibition? In the non-competitive inhibition, the inhibitors bind to site other than active site. So in the case of non-competitive inhibition, the inhibitors bind to the site that is other than active site of the enzyme. Okay, active site of the enzyme. So what we understand by this, that if this is an enzyme, and this is its active site, this is the active site, so the inhibitor will bind something here. So this is where the inhibitor can bind. Okay, but so what we understand that the inhibitor, they do not have shape similar to that of substrate, right? So these inhibitors do not have shape similar to substrate, because if it would have been the case, the inhibitor would have bind to active site, right? Now, what these inhibitors do when they bind to the site other than the active site, these inhibitors, they change the shape of the enzyme. So they change the shape of the enzyme. Okay, because it is a property of the enzyme. When anything comes and binds to the enzyme changes its shape, and this change can either be positive or negative. The positive thing happens when the substrate binds to it, and the negative impact will be when the inhibitor binds to it. Okay, so if anything will bind to the enzyme, it will cause the change in the shape of the enzyme. So in the case of this, the inhibitor binds, it causes a change in the shape of the enzyme, but this change is not positive, and it leads to the decrease in activity of the enzyme. Decrease in the activity of the enzyme. Okay, leads to the decrease in the activity of the enzyme. How? So this change in the shape of the enzyme can be also, this shape can also be the shape of active site. Shape of active site. Okay. So if the shape of the active site gets changed, the substrate will not bind to it, right? And hence it will lead to the decrease in the activity of the enzyme. Now, in this case, if we talk about the enzyme number, so what we can see that the enzyme number or the shape, both. So there will be change, right? The enzyme number will reduce and there will be change in the shape, right? So enzyme number or shape changes, and this change leads to the loss in functionality. This change leads to the loss in functionality, right? So if the enzyme number or the shape of the enzyme change, there will be loss in the functionality, and hence the maximum velocity or the maximum rate the enzyme can reach will also decrease, right? So the Vmax will decrease. Now, if we talk about the substrate concentration, if we increase the substrate concentration, can we overcome this inhibition? The answer is no, because in this case, the substrate is not playing any role. Here, the inhibitor that is binding to any site and causing the change in the shape of the enzyme is playing the major role in the inhibition, right? So the substrate concentration will be unchanged, and hence the Km will also be unchanged, right? So in this case, in the non-competitive inhibition, Vmax will decrease and Km will remain unchanged. Km will remain unchanged. This is important. Now, if you want to plot this, if we want to represent it graphically, how will we represent it? So in this case, the first scenario is when there is this is when there is no inhibitor. When there is no inhibitor, right? So this is the Vmax that the enzyme can achieve. This is the Vmax that the enzyme can achieve, and this is the half Vmax. This is the half Vmax, and this is the K. When the when the inhibitor is not present. Now, when the inhibitor comes into the picture, how would the graph look? So the graph will look something like this. Okay. So although the same, so it will have same Km, the Km will be same, but the V test max, that is with inhibitor, so the Vmax will reduce while the Km will remain same, right? So this is how the graph looks. This is the substrate axis and this is the velocity axis. Now, let's talk about the examples of non-competitive inhibition. So, about the examples. The first example is about cytochrome oxidase. First example is cytochrome oxidase. Now, what is cytochrome oxidase? It is an enzyme. Cytochrome oxidase is an enzyme that is used in electron transport chain. In electron transport chain. So when we are studying about the mitochondria, so these enzymes are present in the mitochondria and they help in the ATP production and respiration. Okay. So the cytochrome oxidase is an enzyme in electron transport chain. Now, what is the inhibitor of this enzyme? The inhibitor of this enzyme is cyanide. Okay. So the cyanide goes and binds to the cytochrome oxidase, but not at the active site, but at any other site, and then disrupt the shape of the cytochrome oxidase, and hence its substrate cannot bind to it. Okay. And apart from this, so what does cyanide do? The cyanide binds to site other than active site. Site other than active site. Okay. It binds to site other than active site, and this leads to the change in the shape of enzyme. Change in the shape of enzyme. Okay. Now, apart from this, there is one more example, and there is one inhibitor called nerve gas. And this nerve gas, where it binds to acetylcholine. So acetylcholine, sorry, one second. So it binds to acetylcholine, and acetylcholine is a neurotransmitter. So that is used to send signals. Okay. So it binds to acetylcholine, and this acetylcholine is neurotransmitter. Okay. Neurotransmitter. So the nerve gas is the inhibitor for acetylcholine. Similarly, the inhibitor for cytochrome oxidase is cyanide. So these are the two examples of non-competitive inhibition. That means that they bind to the site other than the active site. So this is all about the enzymes and the enzyme inhibition. Now, before moving forward, we'll solve few questions about it. Then we will study about carbohydrates. So the first question is, which of the following best describes the class of enzymes that break bonds by generating a new double bond or ring structure rather than hydrolysis or oxidation? Okay, so we were studying about the nomenclature of enzymes, and we saw that the most widely accepted nomenclature is by IUB, International Union of Biochemistry. So the nomenclature that is most widely accepted is by IUB, right? And it gives 6 class of enzymes. And the way or the acronym to remember this is OTHLIL. Okay. So now let's look at the question. The question is saying that this class of enzyme break bonds by generating a new double bond or ring structure rather than by hydrolysis oxidation. It means that it is breaking bond, but it is not utilizing water or electrons, right? So what class it will be? So the first is ligases. So what does ligase do? Ligase, it forms new bond. Ligase, it forms new bonds, right? So it does not break bond. The second is isomerase. The isomerase also does not break any bond, but it leads to the formation of isomers. So it leads to the formation of isomers. The third option is transferases. So what does transferases do? It transfers the functional group, right? It transfers functional group. And the fourth option is lyases. So if you remember, the lyases are the one that breaks the chemical bonds. So if you have CX plus C dash Y, so what does this lyase do? they break these bonds, and they lead to the formation of this double bond and X plus Y. Okay, and they do not use water. So this is the correct option is lyases. Now the second question is, an enzyme that facilitates a process is known as catalyst, right? What does a catalyst do to the reaction regarding free energy to encourage it to proceed? So we know that the enzyme is a catalyst, and since it is isolated from the living system, the enzyme is also known as biocatalyst. Now, what does the catalyst do? The first option is, catalyst increases the reaction rate by reducing the free energy of the transition state, which lowers the activation energy. So the second option is, the catalyst increases the amount of energy contributed to the reaction through heat from the reaction environment, thus increasing the rate of the reaction. The third is, catalyst induces a global increase of entropy to the product state of the reaction, making it more favorable and thus occurring at a faster rate. And the fourth is, catalyst increases the rate of reaction by decreasing the free energy of the transition state, which increases the activation energy. So if you remember, what does this enzyme catalyst do? It decreases the activation energy. First of all, it decreases the activation energy, right? And second thing, what was the activation energy? It was the energy difference between transition and substrate energy, right? So the first thing we'll have to see is the option which consists decreased activation energy. So we have one option here, that which lowers the activation energy. And how it is lowering it? It by catalyst increase the reaction rate by reducing the free energy of the transition state, right? So if you remember the graph was something like this. This was your energy, right? This was your energy, this was your reaction. So this was your transition state, this is your substrate. So what does enzyme do? It reduces this difference. See, it reduces this difference. So the correct option is option A. Now, the third question is, with regards to the enzyme action, this statement is incorrect. So the first option is, malonate is a competitive inhibitor of succinate dehydrogenase, and this is the correct answer we just saw. Let's look at the other option. So this is the correct answer. Okay, so the question is asked about the incorrect options. Okay, so the first one is correct. The second is, the substrate binds to the enzyme at its active site. This is also correct. The third option is, the non-competitive inhibitors bind the enzyme at the site distinct from that binding the substrate. This is also correct. And the fourth is, addition of a lot of succinates does not reverse the inhibition of succinic dehydrogenase by malonate. So this is the incorrect. Why? Because this malonate acts as a competitive inhibitor, right? And we just saw how to overcome the competitive inhibition. We can overcome the competitive inhibition by increasing the substrate concentration, right? Or in the case of this, the substrate is succinate. So if we increase the concentration of the substrate, we can reverse the inhibition of succinic dehydrogenase by malonate. So the D is the correct answer. Now the fourth question is, which of the following modification to an enzyme catalyzed process will change the Vmax? So in what inhibition we saw the change in the Vmax? The first is addition of a non-competitive inhibitor. The second is addition of a competitive inhibitor. Third is increasing substrate to a supraphysiological concentrations. And fourth, none of these options. So what we saw that the change in Vmax happens when there is an addition of a non-competitive inhibitor, right? And the term supraphysiological here means higher than physiological condition. Physiological like normal body condition, okay? Higher than normal body condition. So in this case, the Vmax change occurs when there is addition of non-competitive inhibitors. So this is all about the enzyme. So we have completed 2 biomolecules, mainly one biomolecule, protein, and among the protein, there is one part that is the enzyme that we have completed. Now we will move to the next biomolecule, that is carbohydrates. Okay, so the carbohydrates, these are the most abundant biomolecules in the biosphere. Okay, these are the most abundant biomolecules in the biosphere. Okay, so these are the most abundant. Now, what are carbohydrates? So, if we look at the definition of the carbohydrates, these are the hydrates of carbon. These are the hydrates of carbon. Okay, these are the hydrates of carbon. So, what do I mean by hydrates of carbon? So, suppose you have a mixture, you have a carbohydrate mixture. This is carbohydrate mixture, carbohydrate, carbohydrate mixture. And if you add concentrated sulfuric acid to it, concentrated S2SO4 to it, what will happen? It will result in the carbon plus water molecules. Okay, so it has this hydrate means water, right? So it is hence it is a hydrate of carbon molecule. Okay, now what is the general formula of carbohydrates? So the general formula for carbohydrates is CnH2ON. Okay, so what we can see from here, from the general formula, what we can see that the number of carbons, because if you see the number of carbons and the number of oxygens are same, right? So the number of carbons and oxygen is similar. In carbohydrate, the number of carbon and oxygen are similar. Okay. For example, if you look through example, the example is glucose. So glucose is a carbohydrate and its chemical formula is C6H12O6. We can see it has 6 carbon as well as 6 oxygen. And if we write it in the general formula, how we can write it? We can write it like this, right? So this is the definition of carbohydrates. But this definition is not widely accepted because later it was found that there are many other carbohydrates. Those are not actually the hydrates of carbon. Okay, and they do not also follow this general formula. So a modified definition was given, and that definition was mainly based on the component that will be definitely present in the carbohydrate, right? So let's see what are the components that will be definitely present in the carbohydrate. So if we draw one structure of glucose, so if we draw a linear structure of glucose, it will look something like this. Glucose has 6 carbon, 4, 5, 6, right? Now it will have OH, H, OH, H, OH, H, OH, H and CS2OH. So this is your glucose. Okay. Similarly, if we draw another structure, CS2OH, this is another structure of one carbohydrate. This is CS2OH. Then this is OH, OH, H, H. And this is fructose. This is fructose. Okay. So what we can see, and in not only glucose and fructose, if we'll draw the structure of any of the carbohydrates, the structure will look something like this. So from this structure, what we can see that these structures have hydroxyl, right? And there are multiple hydroxyl groups. So people gave a modified definition, and that modified definition was polyhydroxy because it has multiple hydroxyl group. And apart from hydroxy, what else it has? It has aldehyde group or ketone group, right? So people give the definition that carbohydrates are either polyhydroxyaldehyde or polyhydroxyketone So this is a modified definition of the carbohydrate, okay? Because it has polyhydroxyaldehyde or polyhydroxyketone. Now we will study about the classifications of the carbohydrate from the next lecture. One more important thing that you can write, because this is the most abundant biomolecule in the biosphere, carbohydrates are also called as staff of life. staff of life. Okay. Because all the life constitute of this carbohydrates, and these are the most abundant biomolecules in the biosphere. Okay. So in this lecture, we will stop here. From the next lecture, we will study about the classifications of carbohydrate and the structure of the carbohydrates. That's all. Thank you.